Hello everyone, my name is Harshita Nekkanti, and today I'm excited to share the research I conducted on understanding how different MECP2 mutation types influence the severity of Rett syndrome in children. First, let's start off with a definition. What is Rett syndrome? Rett syndrome is a rare neurodevelopmental disorder, and one of its most heartbreaking features is that children often develop normally until around their first birthday. Parents usually describe their child learning to sit up, babble, maybe even say a few words, and then gradually those skills disappear. This regression typically happens between 6 and 18 months, and it leads to lifelong challenges with motor skills, communication, hand function, and often seizures or breathing abnormalities. So over 80% of children also develop sleep problems, which affects the entire family dynamic. All of this traces back to disruptions in a single gene, MECP2. MECP2 encodes a protein called methyl-CpG binding protein 2, which plays a regulatory role in hundreds of neuronal genes. And you can think of this as a master switch. In the brain, it helps turn genes on and off, shapes how synapses work, and stabilizes communication between neurons. So even a small disruption in MECP2 can alter entire pathways of brain development, which is why different mutations can lead to very different severities. When MECP2 doesn't function properly, neurons can't communicate effectively, and developmental slows or reverses, as this gene is necessary for gene regulation, synaptic development, and neuronal function. MECP2 mutations aren't all the same. There are several major classes, starting with missense mutations, which change a single amino acid and can sometimes be mild or disruptive. Nonsense mutations prematurely stop the protein. Early truncating mutations stop the protein very early, usually causing severe outcomes. Large deletions remove entire sections of the gene. And lastly, C-terminal variants affect the protein's tail end and tend to be more mild. Because each mutation impacts the protein differently, researchers have long suspected that mutation class might predict severity, but no one had consolidated the data in one place. Rett syndrome affects multiple areas of development, and the symptoms often appear in a very characteristic sequence. After a period of normal early growth, children begin to lose skills between 6 and 18 months. One of the first signs is slowdown in motor development. Many children struggle with balance, coordination, or eventually lose the ability to walk. At the same time, there is a loss of expressive language. So children who may have babbled or used some words stop communicating verbally. A hallmark symptom is the emergence of repetitive hand stereotypes, like hand wringing or clapping, which replace purposeful hand use. Many children also develop seizures, irregular breathing patterns such as breath holding or hyperventilation, and more autistic-like social behaviors. Before my project, researchers had made general observations like truncating mutations often being more severe, but the problem was that the information was scattered across dozens of studies, each using different severity scales or focusing on different symptoms. There wasn't a single study that brought together large pediatric datasets and compared mutation class side by side. That lack of consolidated quantitative data is the gap I wanted to address. So my research asked, how do different MECP2 mutation classes correlate with neurodevelopmental severity in pediatric Rett syndrome? I wanted a clearer picture of which mutation types predict severe outcomes and which predict milder ones. I hypothesized that early truncating mutations and large deletions would consistently be associated with the highest severity. These mutation types disrupt large portions of the protein. In contrast, missense mutations and C-terminal variants tend to preserve more of the protein function, so I expected them to be more mild. For my methodology, I combined two approaches, a systematic review and a quantitative analysis. The systematic review allowed me to gather and evaluate existing research in a structured way using PRISMA guidelines, so I could identify studies that specifically reported both MECP2 mutation types and pediatric developmental outcomes. After selecting the final set of studies, I then moved into the second method, which was the quantitative analysis. Then I took the extracted severity data from each study, standardized the different scoring systems, and used a one-way ANOVA to statistically compare the 5 mutation classes. Using both methods together gave me a strong foundation. The systematic review ensured the data was high quality, and the quantitative test allowed me to find clear, measurable differences across the mutation types. I started with 82 articles, and many didn't specify mutation subtypes or didn't provide pediatric developmental outcomes, so I had to remove them. I extracted data on walking, hand use, speech, regression patterns, seizures, and sleep problems. And the inclusion criteria included, as I previously mentioned, specified mutation type, pediatric outcomes, and original data. For the systematic review, I followed PRISMA guidelines to make sure the search and selection process was thorough and consistent. I started with 82 articles across multiple databases, but many didn't specify MECP2 mutation subtypes or didn't include actual pediatric developmental outcomes. After screening the titles, abstracts, and then full text, I narrowed it down to 10 studies that met all requirements. From each study, I extracted very specific clinical measures like walking ability, hand use, speech development, seizure frequency, breathing problems, and the timing and degree of regression. I also recorded the mutation type and sample size for each study. Even though each paper focused on different aspects of Rett syndrome, pulling these data points into one dataset allowed me to directly compare how each mutation class affects development across more than 3,300 children. This table summarizes the 10 studies included in my systematic review, and each one contributed something unique to understanding severity in Rett syndrome. Some studies, like Grillo et al., with over 800 participants, provided large-scale registry data that helped establish broad trends across mutation types. Others, such as Scheich et al. Or Operto et al., offered more detailed clinical insight into specific symptoms like regression patterns, seizures, or breathing problems. Together, these studies covered more than 3,300 pediatric cases, which gave me a strong data set to compare mutation classes side by side. Even though the studies varied in focus and sample size, the consistency in how they linked mutation type to severity made the overall pattern much stronger. To compare mutation groups, I used a one-way ANOVA. Each study used slightly different severity scales, so I had to standardize the scores to allow fair comparison. This let me test whether the average severity truly differed across mutation classes. This slide shows the ANOVA table I used to compare the severity levels across the 5 mutation groups. Overall, the ANOVA was useful because it doesn't just compare the two groups. It checks whether all 5 groups differ from each other in a statistically meaningful way. In my case, the analysis looked at the standardized severity scores for early truncating mutations, large deletions, nonsense, missense, and C-terminal variants. The ANOVA produced an F value of 42.7 with a p-value way below 0.001, which is extremely significant. This means the difference we see between mutation classes aren't random or due to noise. The mutation type truly has a measurable impact on severity. This eventually proved statistical significance, which was helpful to make my conclusion more credible and allowed me to strategize how future implications could effectively help citizens. This ranking here shows the overall pattern, with early truncating being the most severe, then followed by large deletions, nonsense, missense, and lastly C-terminal variants. What was striking was how consistent this pattern was across different symptoms and different studies. Even though each study measures severity in a slightly different way, the hierarchy stayed the same. These results show strong genotype-phenotype correlation. Knowing the mutation class can help clinicians predict how early a child might lose skills, whether they may walk, how their communication might develop, and what therapies to prioritize. For example, a child with an early truncating mutation may need immediate intensive motor and communication support. On the other hand, a child with a C-terminal variant may benefit from long-term skill maintenance. One limitation I noticed was that the studies used different severity scales, so I had to standardize the data, which can't capture every detail each scale originally measured. Registry-based studies also rely on parent reporting, which may underreport milder symptoms like sleep issues or occasional seizures. In addition, many studies were cross-sectional rather than longitudinal, meaning they capture severity at one point in time instead of showing how symptoms changed as children aged. Finally, some studies group mutation types differently, so I had to go through and recategorize them to fit the 5 groups I analyzed, which may introduce slight inconsistencies. My findings showed that MECP2 mutation class is a strong predictor of neurodevelopmental severity in Rett syndrome. I found that early truncating mutations and large deletions were consistently associated with the most severe developmental outcomes, while missense and C-terminal variants tended to be much milder. These results provided clear quantitative evidence supporting clinical observations, giving families and providers a more reliable understanding of expected developmental trajectories. Overall, my study emphasizes the importance of considering mutation type when evaluating and planning care for individuals with Rett syndrome. Moving forward, it is important to develop mutation-specific early intervention strategies, since different MECP2 mutations can lead to varying severity and symptoms. Establishing standardized clinical severity scales would also make it much easier to compare results across studies and track patient progress consistently. Additionally, integrating genetic information with biomarkers such as EEG patterns or epigenetic markers could improve predictions of disease severity and help tailor interventions more precisely. Finally, these results emphasize the potential for genotype stratified therapy trials where the treatments are designed and tested based on specific mutation types rather than applying a one-size-fits-all approach. These are the key sources included in my systematic review. Thank you.